Assalamu alaikum. On behalf of Ahsan Academy, let me welcome you to a presentation on anatomy of the eye. In this presentation, I will start with the discussion on the general idea on sensation, how the orbit is formed and contents of the orbit. Then we will discuss the gross anatomy of the eye with its location first, then gross anatomy and histology of the histological features of the eyeball and refractory medias of the eye. If we look at the face, we'll find that there is variation of expression. Why this variation is there? Because the nerve stimulation that is reaching to the brain is different. So this registration of incoming nerve impulse in a particular place of the brain is the sensation, which may be pleasant or may be disturbing. These sensations received by some specialized organ in different places of the body that may be on the surface or inside the body. These are the sensory nerve endings or specialized epithelial cells. So this can convert the physical chemical change in environment into an electrical impulse that reaches the brain and gives some sensation. This may arise as these receptors may spread all over the body or different parts of the body, or may be localized to some organs only. So depending on that, the sensation may be generalized or special sensation. So special sensations has a specific organ from where the impulses reaches the brain. The receptors are limited to an organ like vision, the receptors are in the eyeball. For the smell, it is in the upper part of the nasal cavity. So specific areas are detailed there to contain the specific sensation. But touch, this can be sprayed all over the surface or different parts of the body. So that is the general sensation. So we can classify the sensation into general sensation and the special sensation. So general sensations may be pain, touch, pressure, temperature, and these are the general sensations, and special sensations are the vision, hearing and the balance, smell, and the taste. As we have already discussed, the spatial sensations, the receptor organ, receptors bodies are situated in a particular organs for the special type of sense. Like you see, this is the vision, the receptors are in the eyeball. For the hearing and the balance, the receptors are in the ear. Smell, the receptors are in the nasal cavity. For the taste, the receptors are on the dorsum of the tongue. So let us discuss eye, anatomy of the eye in this presentation. So eye is situated within the orbit. So we should know how this orbit is formed, where the orbit is situated. The orbit is situated in the anterior part of the skull. You can see this is the skull and this one is the orbit. So this is enlarged here and we can identify the bones forming the orbit. So these walls are formed, the roof is formed by the frontal bone. This is orbital plate of the frontal bone, and this roof extends into the medial wall as well as into the lateral wall. Now this posterior part is formed by the sphenoid, and the sphenoid extends into the lateral wall, into the floor and into the medial wall. So lateral wall is mostly formed by the zygomatic bone with a contribution from the sphenoid. The floor is mostly formed by the maxilla with a small contribution from the sphenoid bone. and the medial wall from the anterior to posterior formed by the frontal process of the maxilla, then the lacrimal bone, pallar ethmoidal bone, and a small, you can see this orbit, coronal section, this is sagittal section, and the roof is removed. So you can see the orbit contains mainly the eyeball. So the eyeball is situated at the central part of the orbit, and this eyeball is surrounded by different structures, and those are also content of the orbit. These are the muscles. You can see the muscles. This is extraocular muscles that helps in the movement of the eyeball. The nerves that to supply these contents as well as that comes out of the orbit to supply the forehead and other parts. You can see the nerves of that. Optic nerves helps in the vision. Other nerves, that is trochlear, trigeminal has frontal, ophthalmic division of the trigeminal has frontal and lacrimal branches. Arteries, you can see this is the ophthalmic artery getting inside and divides into branches to supply the structures, and these accompanying veins come out of the orbit. One gland is there, that is the lacrimal gland. You can see this is the lacrimal gland, and here you can see the lacrimal gland, and its duct systems are within this orbit. And you can see the fascia covering, and the ligaments are there to hold the eyeball in position, and large amount of fat fills the rest of the part to keep the eyeball in the position and allow the free movement of the eyeball. You can see this is the fascia helping the eye to keep in position. There are some ligaments with this fascia to hold this eyeball in position. This is suspensory ligament, and these are the check ligaments, medial and lateral. How this eyeball is maintained in position, already I have discussed that this is having fascia, ligaments, and the rest of the part filled with the fat. So you can see this framework formed by the fascia and the ligaments. The ligaments are the medial and the lateral check ligament. This is medial check ligament, this is lateral check ligament, and the suspensory ligament is this one. Rest of the part, this is the fascia, suspends the eyeball in position and works as havoc. You can see the rest of the part filled with the fat so that the eyeball can easily move, remaining within the position. You can see this orbit, and this is a thin plate forming the floor, another plate forming the roof. So our orbit is very close to other cavities. This is the maxilla and this is the cranial cavity. So this orbit is separated from these important cavities. This is the anterior cranial fossa, and this one is the maxilla, and maxillary sinus is there. So any problem in these areas can spread into the orbit, even and the problem in the orbit can spread into the cranial cavity or go to the maxillary sinus. You can see this is the roof and this one is the floor, and a radiograph showing that this is the tumor in the maxillary sinus spreading into the orbit. You can see this is the area the disease is spreading into the orbit, pressing the optic nerve. What are the features of the eye? Before that, we should be conversant with the terminologies related to the eye. Eye is looking like the world, globe. So this have some common terminological identifications with this eyeball. like anterior pole. You see the center of this layer, it is the cornea, is called the uh, anterior pole. Naturally, the posterior pole opposite to this one, and axis is the line that passes perpendicular to this corner. This is curved, so multiple line can be drawn perpendicular to this, and that line must pass through the center of the pupil. This opening is the pupil. So this is known as optic axis. So multiple line can be drawn as the optic axis. Equator, you see the light, or this is the center part equidistant from each pole, and this line is known as, this region is called equator, and the line joining this anterior and posterior pole is called meridian. So these terminologies are used related to measurement of the eyeball. Let us go for the gross features of the eyeball. It's not rounded, it's having a vertical diameter little less than the anteroposterior and transverse diameter. It's not totally circular ball. This is anteroposterior diameter, and this is the vertical diameter, and you can imagine the transverse diameter. The wall of the eyeball comprises of several layers. Let us label them from outside inward. The fibrous coat. The outermost coat is the fibrous coat, and it is complete and known as posterior. Greater part is the sclera, and anterior smaller part is the cornea. The second layer is called vascular layer, also called pigmented layer. It is named as uveal tract. It comprises of three components. Choroid is the larger posterior part, intermediate part is cilia, and anterior part is the iris. The innermost layer is the nervous coat. That is the sensitive layer, contains the receptor cells. It is named as retina. Now let us go for the content present within the eyeball. So inside the eyeball there is fluid materials as well as lens and relatively semi-solid materials. This is the inside area is divided into anterior chamber and the posterior chamber by the lens and ligaments attached to the periphery of the lens. So anterior to the lens is anterior chamber, and it contains a fluid known as aqueous humor, and this is the anterior part and this is the posterior part of the aqueous humor chamber. This you can see the anterior part, and this small part is the posterior part. This is the lens. In front of the lens, iris, behind the iris is the posterior part, and this is the anterior part. So this fluid circulates through this pupil and goes to the anterior part, and behind this part is known as, within the lens is the vitreous chamber containing the vitreous body. Refractory media that allows the passage of light from anterior to posterior. These are the cornea. After that, the fluid aqueous humor. The light passes through an opening. This is the pupil. Then is the lens through which the light can pass, and finally it passes through the vitreous body and reflects over the retina. So these are the refractory media content within within the wall as well as within the cavities. Light can pass through all the outermost coat of the eye is fibrous coat. Already we have discussed that, and it is part of two spheres combined together. This posterior part is part of one sphere, and the anterior 1/5 is part of another sphere, and the junction is known as limbus. This is one sphere and this is another sphere, and the junction is called the limbus. If you look into the limbus, you'll find one venous channel. This is the venous channel. This is known as canal of Schlemm. This posterior part is the sclera and the anterior part is the cornea. This is the canal of Schlemm. You can see in the limbus, means junction of the cornea and the sclera, also known as sclerocorneal junction. So this is the total fibrous outer coat, and the posterior part is the sclera, and this comprises of dense connective tissue which is very tough, and it is having two component parts. The outer covering part posteriorly is the episcleral lamina, which become continuous with the anterior part. This is the conjunctiva that covers the sclera as well as continuing over the cornea. You can see this is the episcleral lamella, and it is anteriorly, this is the conjunctiva, and that conjunctiva become continuous with the epithelium of the cornea. This is inseparable here in the over the cornea. And internal to this is the suprachoroidal lamella, that is remain intimately related to the outer surface of the choroid. The anterior part is the cornea. This is transparent and it's about 1/6 of the total area. It is a bit projected and the diameter is less than the diameter of the sclera. There is no blood supply and lymphatic vessels for this cornea. It is well innervated and sensitive. This is transverse diameter is greater than the vertical. This you can see the transverse diameter. This is a bit larger than the vertical diameter. So this cornea is not totally a rounded structure. It transversely elongated, slightly oval in structure. The thickness also varies. At the center it is half centimeter, but peripherally this is a bit thicker, it's 0.8 millimeter. Eye is the organ of vision, and this cornea plays an important role in visual function by providing major refractive, it is the major refractive components, and it allows passage of light to the lens and pass through the vitreous body and reflects over the retina. This is an important barrier for that protects the eye and is very sensitive, can initiate some reflexes to protect the eyeball from the physical injuries. Histologically, it comprises of several layers. We can start from outside. Outermost is the epithelium. You can see this is the outer layer, and this comprises of epithelium, which is multi-layer stratified epithelium. You can see this is the epithelium, and gradually cells have become flatter, but it is not squamous, it is cuboidal in nature, the superficial cells. So it is stratified cuboidal epithelium. This lies on a non-cellular limiting membrane. This is known as the anterior limiting lamella or Bowman's membrane. It's a very strong lamella and has some protective role. After that, the thickest portion is there and known as stroma or substantia propria. This is a connective tissue part and this contains specialized ground substance allowing the passage of light, making it transparent. The posteriorly another non-cellular lamella is there. This is known as posterior limiting membrane or posterior limiting lamella. This is also known as Descemet's membrane. So anterior limiting membrane is called Bowman's membrane and the posterior one is called Descemet's membrane. In between lies the main component, the stroma. The posterior chamber, the anterior chamber is separated from this cornea by a thin layer of epithelium. This is simple. So we have covered the outer coat, which is fibrous in nature and comprising of sclera and cornea. The second layer is vascular pigmented layer, also known as uveal tract. You can see here this comprises of choroid, ciliary body and iris. Let me show it here. It starts from the posterior. This is the choroid, ciliary body and iris. So this is the choroid, this one is the ciliary body and anteriormost part is the iris. So these are the three component parts of the second layer from outside. This is vascular or pigmented layer, also known as uveal tract. It is the clinic. The vascular coat is the posterior major part. You can see this is highly vascular cellular coat. It has a central opening anteriorly and you can see this is the pupil colored yellow and it composed of the choroid, ciliary body and the iris. So this is the choroid and this is the ciliary body and this one is the iris. Choroid, it's a very thin, highly vascular and very dark in color and it covers about posterior 80% area of the middle core and is firmly attached to its deeper retina by a non-cellular lamella that is also known as Bruch's membrane. And this tightly adheres the choroid with the retina and retina can usually remains fixed to this and cannot be separated normally. The sclera, you see, this is the optic nerve. It is passes through the sclera and at this point at the margin this is known as optic disc and at this margin the choroid is tightly adherent to the sclera. This choroid comprises of two layers, suprachoroidal lamella and so suprachoroidal lamella, if you consider this one, this is the choroid and outer part is the suprachoroidal. You can see the darker cells are here rich of melanocytes, that is pigmented cells, and it is thicker part of the choroid. And capillary lamella is inside and there are reach of microvasculatures and this is responsible for nutrition of the adjacent part. Choroid is the middle layer and remaining there it prevents entry of light through any part of the eyeball other than the pupil. So it allows only the passage of the light through the pupil and the light getting inside through the pupil are absorbed. If it had been not absorbed and reflected, visions will become blurred. So clear vision depends on the absorption of the light by the choroid. Choroid is an important source of nutrition. Already we have discussed that choroid comprises of pigmented layer and inner part is very vascular. So this vascularity enables the layer. Ciliary body is the second component of the inner second layer, middle layer. So this is the ciliary body, and it's a mass of muscle. It's a bulk of muscle is there, and it makes a ring. So it surrounds the total eyeball area anterior to the choroid. Its base is there. This is the base area, and rest of the area projects and makes some projections. This base forms the ring, posterior part, and this part is suspended in a finger-like projections. These are the ciliary processes on the surface of the ciliary body. You can see this is the ciliary body, and ciliary body has ciliary processes, and on the ciliary processes it's covered by cells. This is bilayer cells. So it is comprising of connective tissue and smooth muscles. These are the smooth muscles, ciliary muscles. And the surface has ciliary epithelium. Iris is the anteriormost part of the middle cord, and it is forming a diaphragm, means it separates a cavity, and it is adjustable opening inside it. So this is known as pupil. The pupil can be enlarged, can be made smaller because of the relaxation and contraction process of the muscle present in the iris. Pupil is usually 1 to 8 millimeter in size, and it lies between the cornea and the lens. You can see this is the ciliary body, and this is the iris, this is the lens, and this one is the cornea. The total cavity is the chamber, anterior chamber. This chamber is the chamber of the aqueous humor. It is split into two as a diaphragm by this iris. So this is totally immersed in a fluid called the aqueous humor. Knowing structure of the iris is important to understand the function. You see, iris comprises of smooth muscle in the stroma of connective tissue. The smooth muscles are of two types, constrictor pupillae and dilator pupillae. You see, these circular muscles are constrictor pupillae. It circulates arranged circularly around the pupil. So whenever this muscle contracts, the pupil size decreases, becomes smaller. Now, these radiating muscles are dilator pupillae. So these muscles, when contract, the pupil size enlarges. The anterior surface, you see, this is the iris, and its anterior surface, there is no epithelium. It contains cells that are pigmented melanocytes, pigment containing melanocytes, as well as connective tissue cell fibroblast. Its posterior surface is covered by the cells that is continuous with the cells of the ciliary processes. These are the ciliary body, ciliary processes there. So far we have discussed the outer layer, the fibrous layer comprising of sclera and cornea. The second layer is from outside, the vascular pigmented or uveal tract comprises of choroid, ciliary body, and iris. Then is the innermost cord, that is known as neural layer, and it comprises of retina, and that continues anteriorly as epithelial non. You see, this is the outer fibrous cord. It comprises of sclera, and this anterior part is the cornea. The middle cord I am showing from the posterior. This is choroid, the ciliary body, and iris. This is the vascular cord, and the innermost cord is retina. You can see this one, the yellow color area. Retina is the innermost cord, and it is the nervous cord. and you can see this is covering more than a half. Two-third area is covered, this nervous layer sensitive to photo reflections, and this is thicker part, but it gradually becomes thinner and become non-nervous at a margin. This is known as ora serrata, from where the neural element discontinue, and this part you can see this is the ora serrata line. Here the nervous element disappears and it continues as two layers of epithelium anteriorly. You can see this is the part, so this area is bilayer of cells, two layers of cells. It continues over the ciliary process, already we have discussed that cells is there, and posterior part of the iris up to the pupil. So we already discussed that this epithelium is continuation of the ciliary process. Actually ciliary process epithelium is continuation of this bilayer. To understand this, embryology of the eyeball should be understood. This is the bilayer cells over the ciliary process. So let us discuss in short how the eye is developed. You see, this is ectodermal thickening, the lens thickening, and from the neural tube and diverticulum arises. So this diverticulum continues and become cup shaped, and a lens vesicle continues. First it was invagination, then separation from the surface. So this is the lens. It is from the ectoderm, it is coming inside. So a cup shaped area is the diverticulum of the neural tube is coming. So whenever this is cupped by the lens, there is one layer and another layer. So two layers are visible here. So inner layer, it is the nervous, converted into the nervous part. You can see this is the lens area getting inside and becoming this one. So this part you can see has converted into the nervous layer, and outer part you can see this is non-nervous epithelial part. So it continues and continues. Here at ora serrata this inner layer ceases and the cell continues, and this is over the inner surface of the ciliary process. So ciliary process, and these are the condensation of the mesoderm. This is the structure of the retina. This is inside, and this one is the choroid. So choroid, you can see very vascular pigmented layer, and then retina starts. Two layers of biembryology. We have understood the outer part, you can see this epithelium, and inner part, this is the nervous area. So a single layer of epithelium is there, and junction, this is the nervous part. So composed of epithelium and neurons, glial cells. So within this there are some modified epithelial cell and forms the receptors, and there are some neuron cells we will get in the retina. Let us discuss in detail structure of the retina. In a biomicroscope, we can identify 10 layers of retina if we enlarge the power. You can see this is the histological features of the retina showing 10 microscopic layers. These from inside outwards, these layers are named here. This is the first layer, is layer of pigmented cells. You can see a layer of pigmented cells are there. The indicator is showing. The second layer is layers of rods and cones. You see this is pigmented cells, then these are the receptor cells, rod cells and cone cells. The indicator is showing. So third layer is limiting membrane. You can see this limiting membrane is there. The fourth layer is outer nuclear layer. This is, you can see many nuclei are there, outer nucleic layer. Then 5 is outer flexiform layer. This one is the outer flexiform layer. Then again there are so many nuclei. So inner nuclear layer and inner flexiform, relatively pinkish in area. Eighth layer is the ganglionic cell. You can see the large cells here. These are the ganglionic cell layer. And the ninth layer, the fibers, means the nerve cell processes converts here as a nerve cell layer, and the innermost is a very thin limiting non-cellular layer is there. So these are the 10 layers visible under high powered microscopes. A laboratory microscope is enough to identify this. Two types of receptors are there in the retina. This is rod cells and cone cells. Figure, you see, this is the cell portion that remains in the outer part, nucleus, and the cone and rod remains in the inner part, closer to the inner surface. So this, as per the shape of this part, it is named as cone, and this is named as rod. So rod cells are sensitive to low intensity light, and this is called scotopic vision. And this is sensitive, the cone is sensitive to high intensity, and photopic vision is the conic vision. This is rod is responsible for night vision. At night, after adoption, we can see objects. This is because of the rod cells. And opsin is the chemical substance that is produced by the rod cells, and the synthesis of opsin depends on the vitamin A. So vitamin A deficiency leads to loss of night vision. On the other hand, the cone is responsible for the color vision and daylight vision. This gives us ability to see the object perfectly. In night visions, you will not feel the color, you cannot see the color. Optic disc and macula are the two special areas. Here you can see this is the optic disc and this is the macula. So let us start with the optic disc. The optic disc is not at the center, it is medial and a bit superior to the posterior pole, and this is the entry point of the optic nerve. It is about 1.5 millimeter in diameter and easily visible through the pupil by the ophthalmoscope. This is relatively pale than the surrounding areas and light pinkish in color. The central part is pierced by vessels. This is the central retinal vessels. Then this can be easily seen by the ophthalmoscopic vision. And here there is no photoreceptor cells, means rods and cones are absent in this optic disc area. So we cannot see the object if it is projected over this area only. The second area is the macula lutea. This is fovea centralis is the other name. It is at the posterior pole and oval in shape near the center. You can see this is a bit thinner area. Here the retina is very thin and it is about 3 millimeter medial and 1 millimeter superior to the optic disc area, a bit away from this one, and it is situated at the posterior pole. Here you see concentration of the receptor cells are more and responsible for good vision. Visual resolution is more here. Optic disc, previously I have said that not containing any receptor cells, and it is pierced at the center by the central artery. So rods and cone cells are absent, as well as the presence of the artery occupies some area. So image over this area, light cannot be received, and so that area is known as, this insensitive area is known as blind spot. Artery supply. This is the ophthalmic artery, and it is branch of the internal carotid artery, and this ophthalmic artery alongside the optic nerve passes through the optic canal. Then it gives one artery, this is central retinal artery, that enters into the optic nerve and occupies almost central position of the optic nerve and enters into the eyeball. And already we have discussed, in the optic disc this can be visible and spreads all over the retina. This ophthalmic artery continues and divides, gives several ciliary branches, and that supplies the outer part of the eyeball course and supply the outer part of the eyeball wall. You can see these are the ciliary artery penetrating the sclera. It breaks us in the vascular layer of the sclera as well as choroid. This is central retinal artery going inside up through the optic disc and spreads over the retina. Central retinal artery is clinically important, and you can see this is going inside, spread all over the retina to supply the retina. This we can easily see by ophthalmoscopic examination, and this is called the fundus of the eyeball, and here optic disc can be visible and arteries can be easily visible. Several diseases affect these blood vessels, like diabetes mellitus. There will be change in the vascular, and by ophthalmoscope easily early diagnosis is possible, that changes has started appearing, and patient can be warned and can take precaution measure. The eyeball is supplied by both autonomic and sensory supplies. The spatial sense as well as general sensations are there. So parasympathetic, it arises from the oculomotor, with an oculomotor nerve, and these are the preganglionic fibers. They ends into the ciliary ganglion. You can see this is the ciliary ganglion. Within the orbit, close to the apex of the orbit, it is situated. This is the ciliary ganglion, and it supplies, the postganglionic fiber supplies the sphincter pupillae. Already we have discussed that contraction of the sphincter pupillae causes reduction in the size of the pupil. This is constrictor pupillae. Then ciliary body muscles are also supplied by this and induce contraction. The contraction of the ciliary body muscles causes relaxation of the suspensory ligaments of the lens, and that causes increase in the curvature of the lens. Sympathetic fibers distributed to the eyeball through the long and short ciliary nerves, and this is responsible for the innervation to the dilator pupil. So they dilates the pupil. Contraction of this will increase the size of the pupil. The sensory supply, the vision, is through the optic nerve, and general sensation. This is the eyeball, this is the wall, and this eyeball contains cavities. This is known as chamber. This is the aqueous humor chamber, and this is the vitreous humor chamber. So this is anterior part has a fluid containing chamber, and this is posterior part, semi jelly like substance are there. This is the iris that is divides this chamber into posterior part and anterior part. First, from the posterior part, you see this is the anterior part, this one is the posterior part. The posterior part is in front of the lens and behind the iris, and the fluid aqueous humor is formed here, circulates through the pupil and goes there to be absorbed. I will discuss in detail in subsequent slides. Refractory media, already I have mentioned that this is having wall and cavity. So refractory media are part of the wall. This is the light ray passing through the part of the wall, fluid part, then this is the pupil, a gap in the opaque layer, and then it passes through some other refractory media to the retina. So in sequence, these are this cornea, then we'll get the aqueous humor fluid part, then this is the lens, and finally this is the vitreous body present in the posterior part, and the light can pass through this and refract it up to produce an image over the aqueous humor is the fluid that is present in this area and anterior part. This is the ciliary processes, part of the ciliary body. The ciliary process covered by the cells and contains very rich capillaries, and from here the fluid aqueous humor is produced into the posterior part, and these circulates through the pupil to the anterior part, and this anterior chamber, it goes to this fluid goes to the iridocorneal junction. This is the limbus, and here this is the iris and cornea. This angle is there, iridocorneal angle, and at the limbus there is a venous channel called the canal of Slane. So fluid passes through this to the venous area and returns back to the blood circulation. It's like lymphatics. You can see this is the lens, and it's a transparent biconvex lens. You can see this is the lens, and it is surrounded by the ring of iris. This is the iris behind, in front, and behind this is the vitreous body, and it is surrounded by the ring of ciliary process. This is the ciliary processes, and it is attached, the outer margin is attached to the ciliary process by ligaments. This is known as suspensory ligaments of the lens. So this way this is kept in position. So lens is kept in position by the support from anterior, this is iris, behind by the vitreous body, and it is suspended, attached to the, it's a transparent structure, biconvex in nature, and composed of transparent elastic capsule, and inside there is simple cuboidal epithelium present anteriorly, and these cells posteriorly become very long, and it's a special type of cells that fills it, and these are known as lens fiber. You can say this is the cuboidal epithelium anteriorly, but posteriorly the cells become very elongated and special type, and these are thin, flattened, and known as lens fiber. The most posterior part is the vitreous chamber, and a transparent gel-like connective tissue present inside is known as vitreous body. This is composed mostly of water, about 99% is water, and a small amount of collagen fibers, very fine collagen fibers are there, a few macrophages there, and one type of cells, that is hyalinocytes, this is embryonic cells present inside it. This is transport area and work as the refractory media. Thank you very much for remaining with this long presentation.